This is the second part of a water microbiology experiment. Previously, you did the presumptive test, which includes the most probable number experiment. This is the second phase, which is the confirmed test. What you're looking for with the water is whether or not coliform bacteria are present. Coliform bacteria typically are found in the intestines, and so it would be an indication of fecal contamination. Therefore, it's not suitable for human consumption. If you remember, there are three phases for the water testing. There's the presumptive test, the confirmed test, and the completed test. The presumptive test, which you would have already done at this point, that's the initial first stage of looking for lactose fermenters that produce both acid and gas. The coliform bacteria that exist in the intestines typically are gram-negative, and they are lactose fermenters. So that's why first you're looking for the lactose fermenters. You would have inoculated lactose fermentation tubes. You would have determined the most probable number. That's just an estimate. It's not an exact number of how many bacteria in the water sample, but it's just an estimate. If the results were negative, the water is fine to drink, you don't move on. But if the results are positive, then you have to move on to the second phase, which is the confirmed test. In this stage, what you're going to do is inoculate a McConkey plate, which is known as the MAC plate, and an eosin methylene blue, or EMB plate. You will inoculate those plates to determine if the bacteria from the water sample are gram-negatives. Both the McConkey and the EMB plates are selective. They're also differential, but they are selective in that only gram-negatives will grow. So you're going to take the water sample, if you had a positive presumptive test, and now you're going to inoculate these plates, then incubate them, and then go look and see if there's growth. There are some gram-positives that can ferment lactose, and so would show a positive result in the presumptive test. And so this confirmed test would eliminate, was it a gram-positive? We're not really concerned about those, but what you are concerned about are those coliform ones, which are gram-negative. If you get growth on the McConkey and the EMB plate, so you get positive results, then you would move to the third stage, which is the completed test, and that's when you would do a gram stain to confirm that it is a gram-negative bacteria that is present in the water. So in this test, we already did the presumptive test. We got positive results, so we took the water sample, and you would have inoculated the McConkey plate and the EMB plate. As you can see here, there is some pretty decent growth on the McConkey plate, so that indicates that yes, it is a gram-positive. Over on the EMB plate, we did also get growth on there, so that indicates that yes, it is a gram, excuse me, these are both gram-negatives on here. So it's a positive result for gram-negative. On the EMB plate, if you notice, this is also a differential plate. So only gram-negatives will grow. That's the selective part of it. The differential part of it is that there is, in the media, some dyes, and the differential part of it is that when E. coli grows in it, it turns it this metallic, shiny green color. It's very distinctive, and it's very unique just to E. coli. So on this particular plate, we can say from this sample that yes, it was indeed gram-negative. We know that from both the plates. From the EMB plate, it is positive for E. coli. How do I know it's E. coli? Because it has that distinctive metallic green color to it. At this point, because they are positive results, then you would take the sample and you would do the final completed test, which is to do a gram stain to confirm that it is gram-negative. Basically, don't drink this water supply. The water could be used for things like watering lawns, anything, but it cannot be used for human consumption.